Okay. So announcements. I will. If you guys didn't have, I know some of you guys did the quiz already, but since you guys had an exam on Monday, I extended it to Saturday night. I'll have quiz 8 out. It'll be due next week, but we'll see how it goes with quiz 7 being due during the weekend. So we'll play by ear on that one. Again, if you haven't uploaded your presentation and you've already presented, please upload it. That's when I can actually put the grade in. So, okay. Oh. So let's start with presentations.
I'll go.
Okay.
It says host disabled.
Oh, whoops. I really got to remember to do that. Okay, now you should be able to.
Is it sharing?
Yep.
Okay. So my question was, for a species with a diploid number of 60, indicate how many chromosomes will be present in the somatic nuclei of an individual that are A, haploid, B, tetraploid, C, trisomic, and D, monosomic. So for the background information, for each term, they each have an equation you can use to find the amount of chromosomes. So for haploid, we use N. Since there is a diploid number of 60 and the equation for diploid is 2N, we can divide the 60 by 2 to get the number of haploid chromosomes. For tetraploid, the equation is 4N. For trisomic, it has equation of 2N plus 1. Something to know about trisomic is it can be a result of non-disjunction and result in an overexpression of a gene. For monosomic, the equation is 2N minus 1, and it could be lethal in humans. So for my answers, for haploid, I did 60 divided by 2, and I got 30. For tetraploid, I did 4 times 30, and I got 120. For trisomic, I did 2 times 30 plus 1, and I got 61. And for monosomic, I did 230 times minus 1, and I got 59. That's it.
Good. Everyone okay with this question?
Yeah.
Cool.
I can stop sharing?
Yes.
I'll go next.
Okay.
It's not working right now. My screen share, it's saying I'm gonna have to quit the Zoom. So can I come back after it quits and present?
Yes.
Okay, thank you.
Rob, do you want to go now or?
Yeah, sure, I'll go. It's telling me the same thing, that I have to quit and rejoin.
Oh, this is weird.
So I'll do the same.
Okay. I think Tatiana's back.
I can present now.
Okay. Technology does not like us today. Ooh, colorful. Tatiana, you're muted.
Can you hear me now?
Yes.
Okay, I'll go back. Sorry. Okay, my question was, a human female with Turner syndrome also expresses the X-linked trait hemophilia, as did her father. Which of her parents underwent non-disjunction during meiosis, giving rise to the gamete responsible for the syndrome? My background information is that Turner syndrome results from a missing or incomplete sex chromosome. People with Turner syndrome are phenotypically female and they have underdeveloped breast and short statures. Non-disjunction is a cell division error in which homologous chromosomes in meiosis or the sister chromatids in mitosis fail to separate and migrate to opposite poles, responsible for defects such as monosomy and trisomy. The answer I got was that the mother underwent non-disjunction during meiosis, which gave rise to the gamete responsible for the syndrome.
Yep, that is correct. Does that make sense to everyone? Yes, no, maybe so. Okay. Great. Is Rob back in? Oh, yeah. Okay.
I'll start. Okay, so my question was question number 2. It is the condition that exists when an organism gains or loses more chromosomes but not a complete haploid set is known as polyploidy, euploidy, aneuploidy, triploidy, and trisomy. So some background information on this question. So chromosomes are passed down from parents to offspring. Humans have 23 pairs of chromosomes, and it's 23 from each parent, so you get 46 in total. A haploid is one set of chromosomes, and then a diploid is 2 sets of chromosomes. So when I first was doing this question, I decided to just search up what everything was. So when referring to when I first saw polyploidy, I didn't really understand what it was. So I searched up ploidy. Ploidy is referring to the number of sets of chromosomes in a cell or organism. And when you're looking at polyploidy, cells have more than 2 sets of paired chromosomes. In euploidy, cells have one more than one complete set of chromosomes. And in triploidy, fetuses are born with an extra set of chromosomes, which leads to developmental issues. In trisomy, 3 copies of chromosomes are available instead of 2, and that leads to physical abnormalities. So the answer to this question would be aneuploidy, since when you have this condition, you don't lose a complete set of haploid chromosomes. You would only have an extra or a missing one. So for example, if if a human offspring has 46 chromosomes, you would either have 47 or 45 because you're missing one and not a complete set of the chromosomes. So yeah, the answer is aneuploidy C.
Great. Okay. Everyone cool with that question? Yes.
Yes.
Okay.
Yes.
Wonderful, wonderful. Let's go back to my presentation. Okay. So as we talked about, well, we didn't really get to talk about it, but I'm not entirely sure where you guys are in lecture. Have you guys started chromosomal mutations?
Started it.
Okay, that's good. So there are two types of chromosomal mutations. One set is called structural, and the other set is just variations in the number of chromosomes, okay? So what do you guys think this is? Can you guys see my mouse?
Is it deletion?
Yes, that is deletion, because you are seeing a loss in like the chromosome itself, right? What about this guy? What do you guys think?
Translocation?
Not translocation. This is the same chromosome.
Is it duplication?
Yes, it is duplication. Does that make sense? It's just this little region over here getting doubled. That's why it's like this way, right? It's showing this sequence is here and here. What about this guy? What do you guys think? So it's kind of twisted around here. What's going on?
Inversion?
Yeah, that is inversion. So inversion is when a set of the chromosome is inverted or flipped around, a 180-degree turn, okay? What about this guy? Okay, I don't know if this one is actually covered in lecture, but this is substitution. It's just substituting a region area, a region in the chromosome itself. And then this guy would be, which is the one we have left.
Translocation?
Yep. So with translocation, it can either be two types, reciprocal or non-reciprocal. So with reciprocal, it's a two-way transfer. Non-reciprocal is one way, okay?
Okay.
So with inversion, there are two types. One is called paracentric inversion and pericentric inversion. Did he get through this already, or
No, I don't think so.
okay, so I'll go over this guy. So paracentric, okay. So with paracentric, okay, we have two chromosomes, right? We have this centromere. In paracentric inversions, the centromere is not involved in inversion. So you see that here you see BCD and then DCB, right? Here. So that's what gets inverted, but the centromere is not involved in it. So it's around it, but not involving the centromere. Pericentric, on the other hand, it ends up changing the lengths of the two arms because it involves the centromere. So an example to this is this guy. where you have the centromere here. Oh, wait. This is where the inversion occurs. You see that there is a difference in the size of the arms here. And you have the centromere in between it. Does that make sense with these?
Yeah.
Okay. So chromosomal mutations, next one we're going to talk about is more of variations in the number of chromosomes. So like Rob was telling us, aneuploidy, it involves the diploid number plus or minus any number of chromosomes. So like in Turner syndrome, that's considered aneuploidy because you're missing one X chromosome. Or like Klinefelter, where you have another X chromosome, right? Does that make sense? So that's a general term for these kind of situations. So each of these guys have, so each of the different aneuploidies, they have different terms, and you can easily figure out which one it is by with the number of chromosomes. So in this case, if you have monosomy, you have 2n minus 1, which is essentially lethal in humans. So with these guys, so you won't end up having any live offspring, okay? Disomy is just another word for diploid. It just stays the same. Trisomy is 1 plus the diploid number. And then tetrasomy, pentasomy, that's just adding another number to trisomy. And then pentasomy is adding 1 to tetrasomy. Does that make sense so far?
Yeah.
Okay. Next, we have a thing called euploidy. Euploidy essentially involves multiples of the haploid set, or N, right? So diploidy, as we know, is 2N. Polyploidy just refers to having more than 2 sets of the haploid sets, okay? So 3N, 4N, 5N, 6N, so on. Then we have, so again, they're also called triploidy, tetraploidy, pentaploidy, those kind of things, okay? Then we have something called autopolyploidy. This is multiples of the same genome, whereas allopolyploidy or amphidiploidy is essentially multiples of closely related genomes, right? Does that make sense?
Yeah.
Okay. So the haploid number in a certain species is 10. How many chromosomes are present in a cell from a triploid member of this species? What do you guys think?
30.
Yeah. So why is that? Because we know a triploid member is 3 times the haploid number. So 3 times 10 gives you 30. Does that make sense?
Yes.
Okay. Which of the following genetic abnormalities is most likely to result in viable offspring in humans? So what do you guys think?
Trisomy 21.
Yep. So trisomy 21 is one of the conditions, one of the mutations that end up with Down syndrome, right? So you definitely see those guys. We know that monosomy, anything monosomy, we're done. Triploidy is not common in humans. You would see that more in like plants and some other animals. I can't think of an example off the top of my head. Haploidy, that's not happening with humans because you're already starting off with half the material. You can't not have the full set. Which of the following embryos is least likely to survive live birth? What do you guys think? Okay, when I said this, I should, okay, what do you guys think, actually?
Monosomy of chromosome 3.
Yep. Does that make sense as to why that's the answer?
I'm a little confused why it's 4 and not 3.
So, for one thing, with it's a little bit different with the sex chromosomes. These aneuploidy situations are more lethal in in the sex chromosome, not sex chromosome. They're more okay. Hold on, let me rephrase. So monosomy is more lethal in autosomal chromosomes because you can see Turner syndrome happening. Like you can have a woman with Turner syndrome. She would just be sterile. Does that make sense?
Yeah.
It's so the question is asking which of the following is least likely to survive to live birth. You are seeing someone who has monosomy X because you are seeing a woman or a female with this condition. Whereas with, especially with the autosomal chromosomes, there are important genes on both chromosomes in the autosomal chromosomes that is required for surviving. So that's why it ends up, monosomy ends up lethal because you're missing part of the chromosomes that an entire chromosome that has the essential genetic material that you need. Okay. How is the chromosome complement in an individual with monosomy written?
Two n minus 1.
Yep. So that's just remembering what's going on with monosomy. Okay. Okay. Which of the following terms refers to a failure to separate a pair of homologous chromosomes during the anaphase 1 of meiosis?
Non-disjunction.
Yep. Yay. Okay. Which inherited disorder can be produced by a Robertsonian translocation? So that's just essentially a translocation between two different chromosomes. Do you guys know this? Or No?
Not really.
Okay, so this is just familiar Down syndrome. I actually had the slide in the video that I'm using for quiz 7. So Down syndrome can occur in 2 different manners. And one of them is a translocation between chromosome 14 and 21. And then you also have the trisomy version of chromosome 21 to actually cause Down syndrome. Does that make sense? Yes, no. Guys?
Yeah.
Okay.
Can you repeat that, please?
Okay. So with Down syndrome, there are 2 ways to inherit it. One is a translocation between chromosome 14 and 21, or you can get it having a trisomy or 3 copies of chromosome 21.
Okay.
Okay, good. Good, good. I don't know how much of the diseases you guys have gone over in class, but how is this guy caused? Or which one is oh, okay, I gave away the answer in the figure, but a small deletion in the small arm of chromosome 5 is known to cause blank syndrome.
It's Credo chat.
Yes. Do you guys know why it's called Credo-Shat?
Yeah, I think we talked about it in class today.
It's amazing. So essentially, Credo-Chat is in English means a cat's cry. It turns out infants that have Credo-Chat syndrome end up crying like a cat's cry. That's why it's called Credo-Chat. Amazing how that it is. But are we cool with a crying cat?
Yes.
Okay.
Yes.
So which of the following is not an example of aneuploidy? What do you guys think? So the first one, we have 2 extra chromosomes in an organism whose haploid number is 10. Choice 2 says an extra haploid set of chromosomes. 3 says 2 fewer chromosomes than the total diploid number in an organism whose diploid number is 20. 1 extra chromosome in an organism whose haploid number is 10, 1 fewer chromosome than the total diploid number in an organism whose diploid number is 20. What do you guys think?
An extra haploid set of chromosomes.
Yep. So aneuploidy, it's just like all of these guys, all of the rest of ones are examples. It's not an entire set that's being added or lost. It's more one or two or like a handful of them, or depending on that set itself, okay? But less than an extra set of haploid, extra haploid set. Does that make sense? Okay. So I'm gonna touch on a little bit of the next topic just because for people who are presenting next week, I'm not gonna go too in depth just because that will just throw you guys. I don't want to confuse you guys more and don't want to go completely ahead of the lecture. So so this is the central dogma of biology. There is a lot more to this than what we're seeing right now. But we're gonna focus on this set where it says DNA makes RNA via transcription, and then RNA makes protein via translation. Depending on things, you can go from RNA back to DNA through a process called reverse transcription, and then DNA can replicate itself to make more copies, right? So please remember this like forever, at least if you are a biology major. Remember it forever. It will come to haunt you. I don't want to get yelled at by the molecular bio TA when she has questions like this on her quizzes and they don't know it and people don't know it. So please remember it. Okay. So a couple of things that come with this central dogma is the differences between like how it's run in prokaryotes and eukaryotes, right? So in prokaryotes, you have a single, you have your DNA, it gets transcribed and translated all together. Like it can happen simultaneously. Whereas in eukaryotes, there's so many other things going on, like you have the addition of introns. In prokaryotes, you don't have introns. So when this comes into play, so the first thing you would do is transcribe it. You have the primary transcript then, and then you have to like get rid of all of the introns because the exons are the coding parts. We do not know exactly what the function for introns are, so we just kind of in the eukaryotic cells, they splice it out. So that's what they do. They process this RNA through 3 little methods called 5' capping, RNA splicing, and 3' polyadenylation. I promise, don't get freaked out. We'll cover this more soon enough. So essentially, they do that processing. They turn into the full mature RNA, and then they get exported out of that nucleus. And then that mRNA goes to a ribosome to get translated, and it becomes protein. So essentially, we're just looking at the differences between what's going on. And these in eukaryotes, transcription and translation cannot occur together, whereas in prokaryotes, you can occur together. Does that make sense so far? Yes, no, maybe. Did I lose everyone? No?
Can you repeat that? I'm sorry.
Wait, which part? Or the entire thing?
The entire thing.
Okay. So what we're looking at here is the differences between prokaryotes and eukaryotes within this central dogma, right? So in prokaryotes, it's just the DNA. Everything here is coding, and you get transcribed and translated. Like once a mRNA is produced, it goes directly into protein, and this will just continue transcribing. Does that make sense so far?
Yes.
Okay. Now with eukaryotes, translation is separated from transcription because transcription and RNA processing occurs within the nucleus. So in here, all of this is going on in here. RNA processing exists because we also have introns in between the coding regions or the exons, right? So we have transcription and RNA processing in here, and then translation occurs outside. Are you with me so far?
Yes.
So what I was saying is that with RNA processing, it's just these three steps combined into one set here. So once you transcription occurs, you get the primary transcript, the RNA transcript. Then that's like, okay, let's do this. We got to get out all the introns, cap it and protect it, and polyadenylate it for preparing for translation. Once that mRNA is completely ready, the nucleus pushes it out into the cytoplasm, and that's where translation occurs. Does that make sense? So it cannot occur simultaneously, like right with each other. Does that make sense?
Yes.
Okay, great. I promise when we go through the steps in more detail, it will make 10 times more sense. But I just wanted to make sure I covered this so whoever's presenting next week has some info to go on. Okay. So one of the next things I want to cover is the genetic code table. What do you guys know about it?
All those are codons?
Yes. So reading a genetic code table, it starts, so this is how you would see with each of the codons, right? You would see it all. They scientists in who was trying to figure out all of this realized, okay, so they were attempting to figure out if it's one, depending on, okay. So with this, they were like, okay, how do we know which one is, how do they code for proteins, right? So in these cases, they were like, okay, so they tried different insertions and deletions of like 1 base pair, 2 base pair, 3 base pair, 4 base pairs. and they were like, okay, so which is the one that is showing a complete difference in the protein sequence, right? And then they were like, they saw this difference in three, with three, what's the word? Okay. When they had an insertion or a deletion of 3 nucleotides, they ended up seeing a difference. They are either missing one or they inserted one, and it's just like poof. So one of the things that you would see is that so the way you read it is, this is the first letter. So if you have like A, and then you have the second letter as U, you come down, you're like U, okay, so we're in this box here. And then if we have, and depending on the third letter, you come in and be like, oh, okay, so if it's G, we come here and we're like, oh, okay, so this is methionine. So this codon is special in that it is also the start codon. So it essentially initiates translation. It also codes for methionine. Whereas, okay, and then these guys here, those guys are stop codons. So they terminate everything, right? So in this case, they have, so these codons don't code for any amino acids. And then all the amino acids you need are all listed with each of their codons. It's degenerate in that, in the sense of one amino acid can be coded by multiple codons. Does that make sense? Are we okay?
I don't understand the theory behind it.
Like, okay, so this will make more sense when we talk more about translation, because it's kind of, okay. So these are the codons that you see in your mRNA. So in this guy, right? So here with the exons, you see when they're transcribed, you see those codons in there, and they're pulling, they're asking the ribosome, they're putting it there for the ribosome to tell you which one is what, right? So it's like, it's, so essentially transcription is so this is like translating things into different languages, right? So some people like me, so I'm trilingual. I know, so for me, I can speak Hindi, Malayalam, and English, right? So with these 3 languages, think of transcription and translation like 2 different languages, right? So I have something going on here that's in Hindi. And I was like, okay, let me transcribe what I'm thinking in Hindi into Malayalam, so I know it even more, right? Because that's one of my languages that I know fluently, whereas Hindi would be one of the harder ones for me, I guess. Then to explain it to someone here, I would translate it into English, right? So that's what this is doing here. So DNA is, think of DNA as a language. The language in DNA is different from the language in RNA. So transcription is pretty much trying to interpret, allowing the components to transcribe it, right? Okay. So we see here that, and then RNA, you wanna, so for any of this to work out, you need to put it in a way that the cell can understand. And the cell understands things by using proteins. Does that make sense?
A little bit.
And then, so this is okay. So this here, this table here is essentially allowing you to help the cell figure out what it's asking for, right? So essentially this code here is in the language of RNA, and it's like one of those decipher codes, I guess. I can't think of the actual word for it, but like it's like it's translating this RNA language into protein language.
How does it do that?
That we will get to in the next session, because that's more on translation side. I just wanted to go over this guy just because I had a couple of questions on the presentations for next week. But does that make sense so far?
Yeah.
I promise you won't be quizzed on this week. Nope. But anyways, it's just the, like, I guess what I want you guys to know is how to read it, and then just remembering key elements of things, right? I'll go, I want to go over a question on this. Oh, okay. Clearly my animations hate me. Okay. So in this question, which amino acids could result from a single base substitution in CCA? So CCA, right? So that's proline. Indicate the base changes that produces each new amino acid. So from the question, we know that we're asking about single base substitutions, right? So from this, you can tell any, so you'll only get, okay, so only the last base pair is changing, right? That means you will see a variation between the row itself and the column itself, right? So that's, so you would only think about changing the first two nucleotides. So in this case, the first one would end up UCA, which codes for serine, because you're changing. So you go through that list and you see that, okay, so we have these three, that are going down the list, right? Serine, threonine, alanine, right? So these are single substitutions of C, the first C. Then these three, we have single base substitutions for the second C, which gives you arginine, glutamine, and leucine. Does that make sense? So you're looking at the code table. When you change a letter, which amino acid you get. Does that make sense?
So why don't we change the A, the last one?
Because so you see here, so we're looking at CCA, right? So this guy is CCA, right? And it's coding for proline. When you change from A to C to U or G, it's still staying at proline. Does that make sense?
Yes.
So it's asking, it's asking for the amino acids that actually occur from a single base substitution. So you can say that proline is one of the, you can put all three of these down and write proline. But yeah, these would be the correct ones because like it's asking about a change or a new amino acid, right? Proline is the same in this case for 4 of them. Does that make sense?
Yes.
Okay, great. Now I am just gonna let you guys watch a video if we have time. Okay, okay, since it's 5:23 and this is like a 5-7 minute video, it's up to you guys. I'll have this in my PowerPoint. Actually, let's stop here. Do you guys have any questions? Concerns?
No.
Just
Where would
about
you,
presenting next week. It helps to know like the theory and why things are the way they are.
I don't remember if I went too much in the theory with that, the questions.
No, it's just presenting. Because if you don't know what things are, then you know, kind of, you know. But okay.
I have a question. I was trying to upload the slide presentation on Blackboard, but I couldn't find where to upload it to.
Okay, so let me open up Blackboard. Okay. Okay, let me share my screen so you guys can see what I am up to. Okay. Can you guys see Blackboard here?
Yes.
Okay.
Yeah.
Okay. So in the course information tab, we have discussion information. This is where you would upload it. The assignment of this guy. So does that answer your question?
Yeah, thank you.
Okay, great. Anyone else? If not, we're done for the day. So have a good week, guys.
Thank you, you too.
You too.
Thank you too.
Yep, not a problem.
You too.